Eye movements paint us a picture of cerebral function. However, studies focused on hand and limb motor function will very often leverage visual guidance as a core of the paradigm, but do not have a protocol in place for concurrent eye movement recordings. This method enables three-dimensional recordings of simultaneous eye-hand movements, which allow us to assess eye and hand impairment in visually guided saccade to reach tasks. Here we will use this to compare patients with chronic middle cerebral artery or MCA stroke to healthy controls.
The objective analysis of eye movements and the characterization of motor control have been proven to assist in disease detection, monitoring, and prognosis in the setting of brain injury, thus serving as techniques to monitor physiologic phenotype. Here, we combine the biomarker potential for a maximized effect. Demonstrating the experimental procedure is Professor Todd Hudson and Dr. Mahya Beheshti from our research laboratory.
Begin by greeting the participant and escorting the participant into the testing room. Then, briefly explain the exam and experimental task. Start the exam by asking the participant to follow the researcher's finger with their eyes while keeping their head in one position. Then, draw an imaginary H letter in front of them and make sure that the finger moves far enough out and up and down, assessing center, up, down, left, right, down, right, up left, and up right. Next, assess smooth pursuit by asking the participant to follow and maintain their gaze on a pencil moving slowly back and forth in horizontal and vertical direction.